Okay, so BC.
Kelly, we're starting.
Oh, Caroline, we're filming. Hi, we are the Hee Haw Productions.
Today we will be talking about the chemistry of the cell.
Cells are the basic building blocks of life.
Every living thing you encounter in life is made up of cells.
In today's episode of Hee Haw Productions, we will journey through the organic compositions of the cell while taking a break at the ion channels and finally reaching our destination at the effects of an unhealthy cell.
We will also feature an egg-exciting lab about the osmosis of, you guessed it, an egg.
Cells perform a multitude of specific chemical transformations that provide the cell with usable energy and the molecules needed to form its structure and perform its tasks. Ions, water, and many small organic molecules are imported into the cell, while cells themselves can also alter these small organic molecules through chemical reactions. Oh, it's time for the life bubble! If cells are the basic building blocks of life, then what makes up a cell? There are 4 different types of organic compounds that are essential to the existence of life. Number 1, nucleic acids. Nucleic acids are the polymers essentially responsible for the transfer of genetic information. 2 types of nucleic acids are DNA and RNA. These 2 compounds make you. Proteins. Proteins are another type of polymer that work in cells required for structure, function, and regulation of your body's tissues and organs. Proteins are also enzymes, catalysts. They work in your immune system. They're in your tissues, in your muscles. They work as something as in your immune system. You eat them. They're in your gastroenterological tract. Basically, proteins are everywhere in our body. Carbohydrates. Carbohydrates are compounds that provide energy to living cells. They can be found in the cell's mitochondria and embedded into its cellular membrane. And the 4th organic molecule is a lipid. The main functions of lipids is to store energy, signal, and perform specific tasks in the cell membrane. We may see lipids as a negative thing, but without lipids, we would all be dead. Just like if we didn't have any of the other organic molecules, but yeah. Polymers are consisted of its simpler units, monomers, and they're held together by covalent bonds. We can break them down using hydrolysis or put them back together through dehydration synthesis. Thanks, Life Bubble. Consider this equation. This equation describes cellular respiration, and without cellular respiration, well, we would all be dead. Now let's take a closer look at this process. In this equation, we see that the products are CO2, H2O, and energy, also known as ATP. And this makes sense because we expend energy and breathe out carbon dioxide. As you can see, the reactant glucose is being oxidized into 2 pyruvate molecules. The 2 oxidized oxygens are forming H2O with another H+ ion. This process repeats itself in every mitochondria in every cell of our body. It is the reason why we are alive.
Sip.
Hi, welcome to Hee Haw Productions. Today we'll be doing a lab on the extraction of our DNA using our spit.
So nucleic acids are primary compounds that are essential to our living. Without nucleic acids, we would not be able to pass down our genetic material to our offspring.
So what we're going to do is we're going to use this piece of transparent glass and we're going to have someone spit on it.
Professional spitter Moonwon, so please spit.
Okay, so the first thing we're going to do is we're going to add a pinch of salt. So hold out your hands, please. We're going to add some salt. Here. So just add a pinch of that. We're going to add just a little more. Yep. Now we're going to add some liquid soap. Okay, now I'll take this salt.
Now we are going to add some grapefruit juice.
Now we're gonna be using Now we're gonna be using alcohol. I'm just gonna add all this to the one pile. Keep doing it.
All right, go.
So we took the cotton part off of the Q-tip and we swirled it into our mixture. And as you can see, the nucleic acid has stuck onto our Q-tip.
It's the white filaments that you see on the Q-tip represent his extracted DNA.
Which means that we can clone him.
Knowing cells are essential to human life. When the growth and function of cells becomes abnormal or unhealthy, major life-threatening conditions may occur. It affects our body's chemical processes, disturbing our body and its state of equilibrium. Let's take a closer look. First off, how do we distinguish healthy cells from unhealthy cells? Healthy cells usually have a clean surface, a sharp cell membrane, and visible internal structures with a microscope. With unhealthy cells, problems with proper nutrition may occur, for only some nutrients may get in the cell and only some waste products may get out. Now that we know about unhealthy cells, let's dive into a rather scary topic. Cancer. There are more than 100 types of cancer where any part of the body can be affected, and here's why. Cancer is caused by changes or mutations to the DNA within cells. The DNA inside of a cell is packed into a large number of individual genes. Each gene contains instructions that tells the cell what functions to perform, to include how to grow and divide. These mutations to a single healthy cell may cause a cell to grow and divide more and more until the growth is uncontrolled. These uncontrolled cell growth forms lumps or masses of tissues called tumors, which can grow and interfere with body systems by altering body function. Another common disease caused by mutations in a healthy cell is sickle cell disease. Let's take a closer look at the life bubble. So first off, what is hemoglobin? Hemoglobin is a red protein responsible for transporting oxygen to the blood of vertebrates. Its molecule is composed of 4 subunits, each containing an iron atom bound to what they call a heme group. 2 of the subunits are called beta chains, and the other 2 are called alpha chains. Thanks, Life Bubble. Now back to sickle cell disease. Abnormal hemoglobin, called hemoglobin S, causes sickle cell disease. The problem in hemoglobin S is caused by the defect in the gene that directs the production of the beta globin in the hemoglobin. Since the normal glutamic acid in the 6th position of the beta chain is replaced by valine, this causes a problem in the beta chain part of the hemoglobin, changing the way hemoglobin functions. It results in altered solubility, altered ability to withstand oxidation, instability, and increased properties for methemoglobin production. In other words, it causes lack of oxygen, fatigue, periodic episodes of pain, frequent infections, and even delayed growth can occur. So these are only 2 of several diseases that are caused by abnormalities and unhealthy cells. As you can see, a single change of an amino acid or a mutation in a cell can cause life-threatening conditions, affecting processes like phosphorylation and cellular respiration.
What are ion channels?
I don't know, Kelly. Can you explain what they are for me?
Yes, ion channels are pore-forming membrane proteins whose functions include establishing a resting membrane potential, controlling the flow of ions across the cell membrane, and regulating cell volume.
There are several processes that are involved in the use of ion channels.
One process is called active transport. This is the movement of molecules across the cell membrane from a region of lower concentration to a region of higher concentration, which requires energy.
An active transport process involving the hydrolysis of ATP to provide necessary energy is the process of moving sodium and potassium ions across the cell membrane, also known as the sodium-potassium pump. Sodium, a major cation in the human body, is important for membrane function, nerve impulses, muscle contraction, and prevents blood clotting.
Potassium, also a major cation, is important for maintaining osmotic balance, membrane function, and is a cofactor in photosynthesis and respiration. This process uses the enzyme referred as Na plus K plus ATPase and is responsible for maintaining the large excess of Na plus outside the cell and the large excess of K plus inside. For example, in this image of the sodium-potassium pump moves towards an equilibrium state with the relative concentrations of Na plus and K plus shown on the left.
Another important ion channel process is called the calcium pump.
Calcium is a major cation that is present in our bones and enamel. It is important for signal pathways, skeletal material, and maintaining potential differences across membranes.
Calcium is in the cytoplasm and is pumped into the endoplasmic reticulum. A hormone-induced ion channels releases the calcium from the endoplasmic reticulum to the muscle cell. Thus, muscle contraction is then triggered by the sudden release of calcium.
So, as you can see, all the processes involved in the use of ion channels contributes to our everyday functions in life. In this lab, we added 3 eggs to a solution of cornstarch, and as you can see, over time, the eggs are becoming smaller and more shriveled up. The eggs are becoming hypertonic. This is because there is more water inside the egg than there is on the outside surroundings, so the eggs are expending water to maintain a state of equilibrium.
So here, rather than placing an egg in cornstarch, we actually placed an egg in vinegar as well. So eggs contain a compound called calcium carbonate, which is what makes the shell, makes them hard, and then vinegar contains an acid called acetic acid. When calcium carbonate and acetic acid combine, a chemical reaction takes place and carbon dioxide is released, which is the bubbles that you see here that are forming. The chemical reaction keeps happening until all the carbon in the egg is used up, and this can be indicated by the appearance of the egg after you take it out. It actually looks rather squishy. The shell looks like it went away, and all that remains is the membrane as shown here.
We also added eggs into 3 other solutions. The egg in the yellowish solution is in highlighter ink, while the eggs in the blue and the red solutions are in food dye. Over time, you can see that the egg in the yellow solution has soaked up most of its surrounding liquid, while the red and the blue eggs have not. So here we place the egg in the highlighter solution under a black light, and as you can see, the egg has completely absorbed all of its surrounding highlighter solution, while the other eggs have not. Let's go to the life bubble.
Again.
Wait, redo that. Wait, go.
Oh, God.
Wait. Now the nucleic acid. Now the nucleic acid. There's nothing.
What is this supposed to do?
Sugar, stop.
No, what is this supposed to do?
Now the nucleic acid should rise up to the top and separate from the other components of your spit.
It's just salt.
Be funny. Sorry.
Why are your hands so
Kelly.
Through the organic compositions of this cell. Gotta get all the hydrogens onto the carbon. We're starting. We're filming now. No, wait. If cells are the building blocks of life,
And cellular respiration.
wait! Cellular respiration! Stop! I was ready that time! Oh, it's.
Sorry. In a cell.
You gotta react to it like carbon. Kill! Kelly, that was so good!
The camera doesn't work like that.
Sorry.
How'd you...
Come spit.
Oh, right now?
Yeah.
Okay.
No!